So what I'm going to do today, I mean, I'm trying to figure out what it's going to talk about a little bit to this group, but I think the important point is to give you an idea of the kinds of things that we do in the laboratory, and we work in a variety of different tumor types. And sort of the broad themes of what we've done for a long, long time is really, we've been very interested in cancer metabolism, we've been interested in transporters, we've been interested in drug development, and I'm sort of going to provide a little bit of an amalgam of that, but talk about some of our work related to epithelial ovarian cancer. And I'll give the kudos to begin with, is that a lot of the driver on this work, at least over the last 3 to 4 to 5 years, has really been with an outstanding graduate student who works with me, Adrienne, who was the person who just spoke. She has, she's been pushing, the envelope, obviously in the context of some of the studies that we've been talking about. So much of this data, not entirely, but a lot of this data really reflects the initiative from Adrienne. So it's basically, I'm going to give a 40,000, maybe a 20,000 foot view of the kinds of the things we talk about. And I know this is a very complex slide. We talk about one carbon metabolism a lot. This is an area that has ebbed and flowed over the area over the last several years, and it is clearly become an extremely exciting area, even more so because there's a great deal of excitement about mitochondrial one carbon metabolism, which is something that we have recently, it looks like we're going to get funded on a new grant on this, and we wrote a recent review that's going to be appearing very soon in Molecular Cancer Therapeutics. The emphasis is really on one carbon metabolism and its compartmentalization between the cytosol and the mitochondria and how these pathways talk to each other. And at the same time, there's become an increasing interest in the focus of this type of metabolism, even in the nucleus. And by the end of this talk, you guys are going to be experts on one carbon 101. We can't talk about one carbon metabolism without talking about transporters. And this is something that I've worked on for a long, long time. These are the major folate transporters that are involved in therapeutics of cancer as well as other diseases. We cloned something called the RFC, the reduced folate carrier, the major tissue folate transporter in the mid-1990s, and described the first cloning of this gene. And we looked at its characterization, its transcriptional, its post-transcriptional regulation, and so on over the years. More recently, there's been interest in other transporters, the proton-coupled folate transporter, the PCFT. This is the GI folate transporter that's involved in transporting dietary folates into the GI. It is also expressed abundantly in many tumors, including epithelial ovarian cancer, and it's important. There's been increasing interest in using this for drug delivery. Folate receptors have received a lot of attention from a lot of groups over the years. We'll talk about this very briefly. This is a family of molecules. They work by their GPI-linked molecules that function by endocytosis. FR alpha is the form in ovarian cancer. There's also an FR beta that's expressed in hematologic disease, and interestingly enough, in tumor-associated macrophages. This is a point that we'll talk about a little bit more. And this just summarizes, in ovarian cancer, FR is expressed extensively. It's actually the poster child for this disease. Companies like Endocyte have been developed targeting this for drug discovery or drug delivery over the years. And the vast majority of ovarian cancer do express folate receptor. And some of the therapeutics that have evolved from this are shown here. Endocyte, which is now part of Novartis, had developed molecules, EC1456 is an example. This is a folic acid drug conjugate that delivers mostly like anti-mitotic drugs into tumors and has shown some level of efficacy. It went through phase 1, and I think this has subsequently been disconnected. I'm not sure Novartis is that interested in folate conjugated drugs anymore. Monoclonal antibodies, farletuzumab by Morphotek. The Immunogen has also been interested in developing molecules as antibody drug conjugates. This is in a phase 1B clinical trial. Onyx developed a small molecule inhibitor. This is in phase 2. It's been around for a long time, but it's an FR targeted agent. So there still remains clinical interest in this topic. One of the challenges when we talk about folate receptor targeting is the breadth of expression. And this is some data that Adrienne generated actually in the lab from primary specimens. I think this is 41 primary specimens from different stages, along with normal O-ring. And the message here is that FR alpha is expressed extraordinarily broadly. It's about a 2,000 full range of expression. So that means targeted therapies that are targeting tumors up here, maybe they're going to be effective, but what about the guys down here? And this is part of the reason we have been interested in using other drug delivery mechanisms, such as the proton-coupled folate transporter, which is expressed in ovarian tumors across the board at relatively high levels by qPCR, also by immunohistochemistry with an antibody that we developed in the laboratory a number of years ago. And we described this in a paper in Molecular Cancer Therapeutics. Returning to 1-carbon 101, well, we talked about the transporters a little bit. So once the folate is in the cells, we talk about the compartmentalization and how this drives 1-carbon metabolism. And this is an acronym for tetrahydrofolate. I've defined it over here, but I'll walk it through so you guys don't really get too confused with all the arrows. Basically, tetrahydrofolate is the reduced form. It's transported into the mitochondria by a specific transporter called the MFT, and in the mitochondria, it reacts with serine, which is derived from cytosolic serine from glucose, to drive the reaction catalyzed by serine hydroxymethyltransferase 2, SHMT2. I'll be talking about this a bit. This is the first enzyme in this pathway. What this does, this generates 5,10-methylene tetrahydrofolate, 10-formyl tetrahydrofolate, and is ultimately metabolized to formate, and formate is transported into the cytosol. Importantly, these pathways, they generate a lot of metabolic products, whether it's glycine from serine. Almost all the glycine in the cell is derived from serine via SHMT2. Derives NADH in the mitochondria, NADPH via aldehyde dehydrogenase in the mitochondria, and it also generates formylated methionine for tRNAs involved in the translation of mitochondrial proteins. The formate in cytosol drives all of our favorite players: thiamidylate, methionine, SAM, and of course purines. All of this ultimately is derived from serine. So the importance of 1-carbon metabolism is huge. The reducing potential here and the glycine, of course, are important for redox regulation, including the synthesis of glutathione. We've been interested in pathways, including purine biosynthesis, that's this pathway right here, as well as mitochondrial 1-carbon. And very briefly, it's a critical pathway in tumors, been shown. The key enzyme, the 1-carbon enzyme, something called GARFTase and aGARFTase, their definitions are down here. They're important therapeutic targets for cancer. They're independent of p53 status. aCAR, or ZMP, which is the product or the substrate of the aGARFTase step, is actually, of course, it reacts or activates APK and inhibits mTOR. And purine loss in itself, if you inhibit this pathway, mTOR is inhibited as well. There seems to be differential metabolism between the normal cells in terms of ability to salvage purines, and there's a frequent loss of purine salvage in many tumors, including the loss of the enzyme MTAP. And finally, there seems to be a relationship between BRCA status and purine biosynthesis. In terms of mitochondria, SHMT2, the first enzyme, and methylene tetrahydrofolate dehydrogenase 2, the second enzyme, are amongst those 5 different expressed genes in cancer, including epithelial ovarian cancer. It's a primary source of formate, which I just mentioned, which drives all C1-dependent biosynthesis. It's a source of glycine, almost all. It's a source of redox potential if required glutathione and thyrodoxine, and to minimize ROS. And it's a source of ATP in this reaction catalyzed MTHFR1. So it generates ATP, and it supports basal respiration through its role in modifying tRNA. So clearly an important pathway. And with this in mind, we decided 3 or 4 years ago that we were going to generate targeted inhibitors for mitochondrial 1-carbon inhibit pathway. And this was partly and for tool drugs to investigate the regulation, interaction, the control of this pathway, but also potentially as potential therapeutics. We had, prior to that time, been developing purine, de novo purine biosynthetic inhibitors, and the lead molecule here was a molecule called 94, which we'll talk about a little bit more. The lead molecule I'll talk about here is mostly this 347. And this is Dr. Ganji, who is my medicinal chemistry colleague, who has been synthesizing these molecules over the years. We have to study transport, and one of the things we have always done is we developed engineered cell lines that have no transporters, and then we put them back in so that we could use this as a biological readout of how these drugs are getting into cells. And this shows what happens with methotrexate, which is a classic therapeutic. It has no selectivity for cells that express the reduced folate carrier or that which expresses the proton-coupled folate transporter. But our AGF94, one of our lead molecules that inhibits purine biosynthesis, shows exquisite selectivity for the proton-coupled folate transporter in this proliferation assay. We measure transport. That's something we've done for a long time. We generate cell lines that are engineered with knocked out transporters. This is the iGrowth cells that we knocked down, folate receptor alpha, and we looked at uptake of one of our lead molecules in a radioactive form. We can see that it is suppressed. These cells also express the proton-coupled folate transporter shown on a Western, and by transport of tritiated AGF154 under selective conditions that favors PCFT. And what we found in this study, again published in 2017, was that this compound, as well as another molecule, 278, they show sensitivity towards these iGrowth cells independent of the loss of FR alpha, demonstrating that PCFT is an important mode of drug delivery. Turning our focus back to mitochondrial metabolism, AGF94 was developed as a specific inhibitor of purine biosynthesis at the step catalyzed by GARFase. We're interested in developing molecules from the second generation with this modification of the purine ring that target SHMT2, but also at the same time multi-targeting GARFase and aGARFase. So we would have a dual inhibition of mitochondria as well as purine biosynthesis. The goal is to inhibit glycine synthesis, redox potential, and formate required for these critical cellular pathways. And this would ultimately result in a loss of purines as well. Because of the similarity between SHMT2 and the cytosolic homolog, which runs in the reverse direction, we likely would be inhibiting SHMT1 as well. We've been very fortunate. We've had a lot of fun. We've been working with X-ray crystallographers. This is a colleague of mine in Indiana. And Charles has actually crystallized 347 with SHMT2 at a high resolution. This is a previous iteration. He's done this with GARFase as well. These efforts, along with enzymology, has really driven our drug development efforts. And this shows inhibition, Ki values for targeting some of the analogs with SHMT2, GARFase, aGARFase, and SHMT1. And you can see 394 is a very good inhibitor of GARFase, allows inhibitor of SHMT2. But you look at 347, basically, is a fairly good inhibitor of all these enzymes across the board. So at least from an in vitro standpoint, this seems to be doing what it's supposed to do. Of course, does it work in cells? And one of the, again, biological assays we've done, we do something called rescue experiments. And this is how you have this work. This was done in KB cells by Carrie O'Connor, a research assistant in the lab. And in this study, we focus initially on AGF94. And we can see that when we look at inhibition of AGF94, it is almost completely rescued by adding adenosine as an end product in glycine and nucleoside-free media. We look at the other analogs that are all purported SHMT2 and purine inhibitors. Adenosine does not completely protect. Glycine doesn't do much of anything, and adenosine plus thymidine or plus glycine completely protect these molecules. And this is exactly what would be predicted for a molecule, an inhibitor that targets SHMT2 and also inhibits de novo purine biosynthesis. So we've achieved our purpose. One of the things that we've been working on for years is we want to confirm what's going on in cells, and we've become actually pretty adept at doing metabolomics, targeted metabolomics. We cut our teeth on this in this area with Joshua Rabinowitz at Princeton and Greg Ducker, who was at Princeton, he's now at Utah. And more recently, we've been working with Jing Li at the Cancer Center. And this has been a great experience. Jing has become a close collaborator. She's actually a co-investigator on one of our grants. This is actually studies that Adrienne spearheaded working with Jing. It was done in SCAV cells, and I'll show you how it works. It's relatively simple. We're using deuterated serine in this. It's a flux analysis. So it's a deuterated serine that has 3 deuteriums attached to it. The way the metabolism works is serine degradation, catabolism is the primary process. It occurs in this primarily occurs in the mitochondria, with SHMT2 as being the driving player here. So we start up here with our trideutyrated serine, and we do work in sort of a counterclockwise fashion. What happens is your deuterated serine is converted to a monodeutyrated glycine and a dideutyrated methylene tetrahydrofolate by MTHFD2. This is converted to a monodeutyrated tetrahydrofolate, formula tetrahydrofolate, and ultimately to a monodeutyrated formate. This passes into the cytosol, and our readout here is ultimately it's incorporated into thymethylate, DTTMP, and DTTMP as a monodeutyratum if it's derived from the serine in the mitochondria. This reaction in cytosol, SHMT1, can run in reverse, but generally it runs in the direction of glycine to serine. If it runs in reverse, this would form glycine and dideutyrated methylene tetrahydrofolate, and you would generate a dideutyrated TMP. So now that we're all experts on the flux analysis, we'll show what our results look like. This is looking at serine, and it's looking at the various forms of the isotopomers, the M+1, M+2, and M+3. And first thing that comes to mind if you look at your SHMT2 knockdown and drug-treated SCAV cells, you can see that the levels of total serine are higher than in non-treated cells. Okay, this is exactly what we expected if you had inhibited SHMT2. For 2 of the 3 compounds, we see a very marked suppression of total glycine. Again, what you expect. I haven't figured out what's going on with 363, but this emulates the SHMT2 knockdown very closely. This is increased, this is decreased. Exactly what you expect with an SHMT2 knockdown. What's also interesting, if you look at the different isotopomers, you see increased levels of the M+3 serine. That's the purple here. And that's exactly what you would expect if metabolism of the M+3 was decreased in terms of the formation of the methylene tetrahydrofolate. Another insight is obtained by looking at this TTP. This is the level where you look at incorporation into this pool. TTP levels are decreased for the SHMT2 knockdown as well as the drug treated. And this is partly because of limiting formate to drive these processes. Also could relate to a direct inhibition, but we believe it's due to limiting formate primarily. But what's interesting here, it's not only decreased, but if we look at the M+1, which is this methylene going to formate and ultimately be incorporated here, it is substantially depressed in the presence of the drug treated and also with the SHMT2, confirming that in fact this pathway is not going very effectively and consistent with the direct targeting of SHMT2. We extended this to other metabolites, de novo purine biosynthesis. For 94, we included that as a control. This is a GARFTase inhibitor, so it inhibits this target here. This would result in increased accumulation of GAR and decreased levels of ACAR, which is right down here, which is exactly what you expect for a GARFTase targeted drug. For the other inhibitors, as well as SHMT2 knockdown, GAR is elevated as well as ACAR is elevated. And this is because of limiting formate and very likely due to secondary inhibition of GARFTase and ACARFTase by our targeted drugs. And as you might expect, there would be a suppression of ATP and IMP from this sequence going from PRPP to IMP and subsequently branched to AMP and GMP. So exactly what we would expect with ACAR and GARVtase. So we've shown that in fact we target SHMT2. We've also shown evidence that we can inhibit GARVtase and ACARVtase. Finally, an important role here relates to redox potential. In fact, as I indicated, SHMT2 synthesizes glycine, also drives the synthesis of NADH, and this is ultimately required for glutathione. Adrienne recently looked at glutathione and glutathione reduced and oxidized levels, and as we can see in SCOB cells treated with drugs as well as the knockdown, we see a significant suppression of these pools. We're expanding this to look at ROS, and we're looking at other metabolites as well in the mitochondria. Very lastly, very briefly, we've done some in vivo studies. We've shown in vivo efficacy with 94. This is with iGrove, not the best model, but clearly shows efficacy. We were fortunate to obtain a PDX model that had generated from Carmanos a number of years ago. It's a high-grade serous PDX that was generated by Lisa Polwin in the core. We see potent efficacy. We also looked at AGF347 with SCOB, and this is showing 7 mice. And the take-home message is that we see efficacy in excess or better than what for us cisplatin, since these cells are relatively cisplatin resistant. More recently, we've turned our attention to syngeneic models, and there's a few reasons for doing this. One is the reviewers are always asking about mice with the intact immune systems. We obtained from Dr. Orsoluk at UCLA a BR-Luke syngeneic model. This is BRCA negative, P53 null, and it's actually proved to be a very outstanding model for doing syngeneic studies. We also wanted to look at the role of the immune system, potentially as the modulator of drug activity, and how we may be able to target the immune system. Our initial studies, this is showing just efficacy. This is a pilot study. We're doing a larger study as we speak. That shows that AGF94 does in fact have efficacy towards the BR-Luke syngeneic mouse model with T/C of 28%. This is showing our initial foray into looking at impact on the immune system. This is in 6 mice. We harvested the mice after 21 days with dosing with either 2 doses or 3 doses of AGF94, and then we looked at the various immune population in terms of tumor-associated macrophages. We also looked at T cells, and the results are summarized here. That in 2 of the 4 mice that were treated with drug, we see a substantial suppression of the FR beta associated macrophage population, something in the order of 70 to 80%, and notably there was no impact on the T cell population. We're very excited about this. We're doing a phase 2 or a second stage trial here on a much larger scale. But this is really interesting because there's been a lot of interest in the role of FR beta expressing macrophages and how whether this in fact could be a target for this class of drugs. And this gets at the role in terms of macrophages, the tumor-associated macrophages, in contributing to an immunosuppressive environment that permits tumors to evade immune suppression. So this is something that we're ongoing. This is the group of folks that have been working on this in the last few years. A number of people from the lab that have contributed in various ways. A lot of the work was done by Adrienne. I can't give her enough credit. Amode, another outstanding graduate student, and outstanding collaborators that I've had. I'll be happy to take questions. I know it was a lot of Metabolism 101, but thank you very much.